Got it? All right. So I'm gonna work this up, and I'll have it ready for you guys on Thursday. I'll spend some time in class on Thursday showing you what I want you to do with it. Sound good? Nobody came to my gig on Saturday.
Who's on
Sorry,
Saturday?
we didn't go on till midnight. Which for an old fart like me is like really late.
Oh, your band? Most
My
people
band.
aren't 21 though.
That's true.
There's probably like less than 10.
Really?
Um,
I can't judge age anymore. I'm really bad at that.
I
Yeah.
would guess.
Yeah, I guess looking around, I don't see a lot of gray beards out there.
I would have went if I was 21.
Oh, thanks,
would-
Garrett. Means a lot.
I'll look you up once I turn.
Actually, one of my former students did come. It was pretty cool.
I'll come.
He's a
to
musician
shit
as
days.
well, so I saw one of his
Off the
shows.
wall.
He came to my show. It was fun. He's like a really good musician. So, you know, it's like, oh, you're here. He's like, yeah. I'm like, oh, thanks. Oh, it was good. It was good. Actually, two of my former students. Another former student of mine. This guy Dio Jean-Baptiste. He's still taking classes here. He is no joke considered to be the hottest drummer in Seattle. Like, he's amazing. He went to Japan and met some cats over there and cut a jazz album with them. Like, the dude is sick. You'll see him around campus.
Yeah, that's super good.
I mean, you wouldn't know it yet. but you'll see him around.
That's pretty cool.
Alright. So if I'm a little like dragging today, it's because it's taking like multiple days to catch more sleep. Okay. All right. I'll go. So, we talked about the soil, the nutrients in the soil, the water in the soil, how plants get that. Now we're going to look at what the plants do with it once they get it into them. Okay? This is what's called transport. Transport. So, remember, plants don't have a heart. They don't have muscles. They can't, use mechanical devices in order to move stuff throughout their bodies, but they're still able to do it. And they can do it phenomenally well. For example, giant redwood trees can grow up to 300 feet tall. And what's amazing about this is they're actually transporting water all the way from the bottom of their roots, which could be 100 feet underground, to the top of the tree, which is 300 feet in the air, right? So 400 feet of transportation without using a single pump. Pretty cool. How do they do that? Okay. So there are two different sort of methods of transport. One is short distance, moving things over short distances, and that, as we're going to see, that's pretty simple. That's pretty straightforward. And then there's this long distance stuff. Let's like all the way up and down a tree. We'll look at that in a second. Let me see here. Okay. So remember the two different tissues that make up the vascular system in plants. We have the xylem, which is for water, okay? And xylem always moves from root to shoot. Very good. The second one is phloem. And this is transporting sugar, sap, other metabolites. And this moves from, remember? Source to sink. All right. So, for example, so here's the xylem moving up here from roots to the shoots. All right. The phloem, though, could be going from the leaves where the sugar is made, where the photosynthesis is occurring, down to the roots, or it could be going from the roots up here. This is showing it going the opposite direction. But it could be moving in either direction. Phloem can even move from leaf to leaf, or from branch to branch, or from root to root. And remember, it's always just going from source to sink. What it does require, though, we'll see this in a bit, that phloem requires the water found in the xylem in order to make it work. Okay, so that's long distance stuff. Let's look at the short distance stuff. Short distance transport. We're talking about passive and active transport like you guys learned about in 211. This should be somewhat of a review. Remember, passive transport is transport across a membrane, and it requires no metabolic energy. When I say metabolic energy, what molecule do you think of, Mohammed? Thank you, Mohammed. Does not require ATP, right? So remember, cellular transport, cellular transport. Active uses ATP, passive, no ATP. Okay. That's, I need to cut these slides. As I've gotten older, I'm just like, I hate words on slides. Do you guys hate words on slides too? I just hate words on slides.
Sorry.
It's just, I think images are just so much, don't you think images are better?
It's better when you already know what it's talking about.
It's better when you know what it's talking
Like,
about.
if I looked at that, I have no clue what's going on.
Really?
But like,
Didn't you guys learn
in
about
a
this in
way.
211? You guys learned about this in 211, didn't you?
the
It's
words
a bit of a
help.
review, right? So this is an example of active transport, right? This is a proton pump. Did you guys learned this, right?
Yeah, I mean, I know it. I'm just saying the words do kind of help.
The words. Do you want to go back? I can go back.
No, it's fine.
Sure. Who wants me to go back? I can go back. Want to go back?
Pictures
It's
are
just a
better.
lot of words.
Pictures are better.
Okay. So what's happening here? Active transport, ATP, is being utilized, right, to help pump protons across the membrane. You guys remember this? Okay. This creates a gradient, right? More protons in this side than on this side. This gradient can then be utilized to harvest energy in some way, to make, for example, ATP, or NADH, or something else. All right. And what's going on here is the shape of the transmembrane protein is changing, and that's allowing this ion to move. All right. Use a little bit of energy. Okay. Once you have this gradient, things will move down the gradient. They'll just flow, they'll diffuse across that gradient. The issue, though, is the membrane itself can become a barrier. So these transmembrane proteins will just create the proper environment so that they can move across, right? They don't actually need any energy because they're moving down a gradient, but they do need the right environment. So for example, if this, this is hydrophilic and the membrane's hydrophobic, they need a hydrophilic pathway in which to move through it, otherwise they can't do that. Okay. Like I said, this should be super. All right. Some other ways that they can make this happen, so this is still passive transport, is what's called symport. Symport is when a substance in a low concentration hooks up with a proton, which is a high concentration over here, and as they hook up together, it allows them to move across down the proton concentration gradient, but against its own gradient. All right, so this is called symport. Remember symport because it's doing a sim with another molecule. Did you guys learn about that in 211? Maybe, maybe not. No, okay. It's just a form of passive transport. Similarly, there's antiport, in which we're moving, again, once again, this molecule is moving against its concentration gradient, but it's doing it as the proton is moving in the opposite direction, right? So it's actually moving in the opposite direction, okay? So there's symport, moving together, and the antiport moving opposite directions. Move together. Antiport moving in opposite directions. Okay. All right. And again, we're talking about here are different nutrients. Yeah.
Wait, so if there's like more of that high or low concentration, like goes through the gradient, and if there's like more of that inside would there be like a gradient that opens and like, you know, like the sodium 1Q plus pump, like how is that different? Is there any here?
Yeah, it's the same basic idea. Yeah, totally. Remember what the plant is trying to do is it's trying to increase the concentration of these soil ions inside of the plant, right? That's what the plant is attempting to do. So it's taking something that's in a low concentration out here, something like it could be, you know, potassium or calcium, something like that, and it's trying to increase the concentration in here, and it does that through different means. It could be antiport, symport. It depends on the nature of the specific ion, and there's so many, I'm not going to go into all of them, but these are just mechanisms that they use. The reason I like to talk about this is the same stuff that you learned about in cell biology, it's just a different utilization, different example of it. Yeah, so the stuff that you learned before comes in handy. Plants are using this too. Okay, all right. So that's short distance movement. Now we're going to look at the long distance stuff. Okay. So this is what's called bulk flow. So bulk is in like a big quantity. Like when you go to the store and you buy like bulk. Anybody shop bulk? Anybody shop? Shop bulk, like bulk rice, bulk beans, some like that kind of stuff. You get like a large amount of stuff. All right. Okay. So we're going to first look at this occurring in the xylem. So the xylem sap, it's mostly water, but it does have some other stuff in it. That's why they're called sap. We're going to go back and revisit the concept of osmosis. Who remembers what osmosis is? It's the fusion of water. Good. It's water down its own concentration gradient. Okay. I'm going to introduce this term, water potential. Yes. Psi. It's just psi. That's the Greek symbol psi. Okay. So. What is potential energy? What is potential energy? Does this have potential energy? Yes, right? It has the potential to do something. Energy is just the ability to do something, okay? We think usually of potential energy mechanically, but it happens chemically as well. And water can have a certain amount of potential energy in it based upon its own gradient. Remember, gradients, when there's a difference in the concentration of something across a distance, whatever that thing is wants to diffuse. And the more it wants to diffuse, the more energy it has, and we say the greater the potential. So for example, if I took all of you and I stuck you in that back corner over there, right? You wouldn't want to stay there, right? What would you want to do? Come over here, right? And I can harvest the energy of you moving down here. I could harvest that. I could take a little like hamster wheel and you run over here, charge my phone for me or something, right? Okay. Well, we're going to see again that water can have its own gradient, and then that gradient has a certain amount of force, a certain amount of energy in it, and that's the water potential. Okay. Now, pure water has potential of 0. Okay. And whenever we add solutes, to what we call the solute potential of water. What happens to the concentration of water when we add something to that water? It goes down. Good. Therefore, the solute potential is always less than 0, less than 30. There are no solutes in it without 0. Okay? So as soon as you add solutes to the water, that solute potential goes down. Water can also be under pressure, and this is what's called the pressure potential. So if you push or press on water, that's pressure. Water can be pulled too, so it can have a negative pressure potential. It can be under compression or torsion, pull on water. Pumps create torsion, right? Or actually, let me take that back. You create torsion, you pull on water when you use a straw, exactly. Right? You create a vacuum on one end in your gut, and then all right. So the overall water potential is equal to that solute potential plus that pressure potential. Both are combined. All right, so let's look at an example of this. This is the classic, everybody's seen this before, right? I've never actually seen one of these in real life, just in diagrams and books. The U-shaped tube with this mysterious selectively permeable membrane, right? Why is it selectively permeable? Because water can pass through, but other stuff, well, the solutes cannot. So, for example, we have pure water on both sides. It's balanced. We add a solute, for example, let's say sucrose, to one side, okay? By adding the sucrose, that lowers the solute potential. We have not added any pressure to it. We have not, pulled on it either, and therefore the pressure potential is 0. The overall water potential is the combination of the two. 0 plus negative 0.23 is negative 0.23. Now, the water potential on this side is still 0. It's negative on this side. Therefore, what does the water do? It moves the other way. Very good. Okay? You guys have seen this before, right? This is classic osmosis. Now, if you want, you could add pressure to this side, and you could force the water through that membrane. Okay? This is how reverse osmosis works. Anybody spend any time on a cruise ship or anything? Or you time on a boat? No? I did my master's in oceanography, and the boat made its own water. Well, it didn't really make its own water. You're in the middle of the ocean. There's plenty of water around. But what did it do? It used reverse osmosis, basically pumps, to push water through a selectively permeable membrane. It pushed the water through, but the salt didn't. So the stuff that came out the other end was relatively pure water. Okay? And like I said, it works either way, either by pushing on this side or pulling on the other side. Either way, you're going to get the same result, which is water's going to move from this side to that side. Does that make sense? So either if I push on this side, I'm adding, say, 0.4 kilopascals to this side, and that combined with negative 0.23, that's a positive potential that's going to move the water that way. Okay? Or I can pull this way, that's a negative 0.4 on that side, and still you've got 0.23 this side, it's going to flow that way. That should be a negative 0.23, by the way, which is still going to flow that way. You guys see that? So remember, water is always going to move down its own water potential gradient, right? It's going to go from areas of high potential to areas of low potential. In the same way that this is going to go from areas of high potential to low potential. Sound good? Questions? I see a lot of blank faces. I can't tell if that's because you're bored or because it doesn't make sense.
So the first one, it should be positive 4.3 for the pressure, right?
Yeah, that should be positive and that should be negative, so the result is 0. Right? And unfortunately, I can't change these things. Yeah. I could, I guess I could go in there with some white out. light out on the screen. Yeah, that should be positive, yeah. Great. Other questions? No? All right. What's that?
Is it possible to have a positive for all these numbers?
You can have a positive, yeah, this is, over here, this is positive. There you go. So 0.4 minus 0.23 gets 0.7. So 0.7, the net water potential on this side is positive. And so this is still 0 here. I'm just gonna push over that way. Okay. All right. Now, what happens in cells? So, the thing about cells is that the water in the cell always has solutes in it. So it's always going to have a negative solute potential. So if you put a cell, a plant cell for example, in pure water, water is going to move in because of osmosis. Now the cell wall will exert a force on that expanding cell, okay? And it will do that up until a point so that it balances out that pressure. So the pressure increases as water comes in, but then the cell wall pushes back on that and it reaches equilibrium. When this happens to plants, we say that it is turgid, meaning that it's full of water. Okay?
But it wouldn't like,
If it gets to be too much, it would, yeah. If it's too much, it'll do that, yeah. That actually happens to animals as well. If you just like, if you spend too much time in like a freshwater lake, like the cells rupture or something like gross. Okay. Now, if we were to put it in a, like a sucrose solution, for example, which has a lower solute potential, the plant will lose water, right? Now, the thing about the structure of the plant cell is it has no way of pulling on the water on the inside. It can resist the push of the water, but it can't resist the pull, and therefore the stuff on the inside will shrink. And then when this happens, the plant wilts. It goes what we call flaccid, becomes flaccid. This happens to like carrots. If you leave them in the fridge for too long, they lose their water and they shrivel. Flaccid. All right. Questions on that? No? All right, cool. Now, when it comes to getting the water into the plant, it has to come in through the roots, via the root hairs. Now, plants have evolved ways in which to sort of filter out the stuff that comes in. And there are 3 different pathways that the water can follow. The first is what we call apoplastic, and it is occurring between the cells. So it actually flows in between the plant cells. It's moving inside what we call a transmembrane space between the cells. And it can do that until it hits what's called the Casparian strip. And Casparian strip is right here. It's this yellow thing here on this model. It's right on the edge of that stele, which is that vascular bundle. Okay? Right on the edge there is this waxy strip that seals off that transmembrane space. And the reason the plant has evolved this is to force whatever water's coming in from the outside through these cells here. And that allows those cells to filter out any potential, harm to a certain extent. It's not a perfect system. Okay? Symplastic, between, what's going on here is the plant is moving water between what are called the plasmodesmata. Plasmodesmata are these holes inside the cell wall. You've got the plant cell like this, and it has these pores. Again, plants use their structure. They don't have a heart, they don't have veins or arteries or anything, they don't have muscles to move stuff. They use the holes between the cells as a way of moving that water. Again, when it hits the Casparian strip, it has to go through the cell, okay? And the cool thing about the symplastic pathway is it passes through these, the cytoplasm, right, like this. The third way of doing this transmembrane, it actually moves through the central vacuoles of the plant, and it just flows right through the whole thing. Okay, it's similar to that symplastic transport, but it actually goes, you can see here it's moving around that central vacuole. This moves just straight through the central vacuole. Okay. All right, and this is just, that shows that Casparian strip here at the edge of that stele. Let's go through that. All right, so, only got 10 minutes left, so let me get to transpiration. So this bulk movement of water through the plant is what we call transpiration. And what's happening here, people used to think it had to do with capillary action. You guys know what capillary action is? Okay, capillary action, you put a piece of paper in water, it sucks up. That's not what's going on. What's actually happening is water is moving down its own water potential gradient from the ground to the air. So the ground is always wetter than the air, right? So there's more water in the ground, and it wants to evaporate out of the ground. What the plant's doing is basically just taking advantage of that gradient between the ground and the air. It's taking advantage of that evaporation. Okay? Now, where is the evaporation occurring in the plant? It's occurring in the leaves. In the leaves of the plant, at the stomata open, water is evaporating from those leaves. Okay? At the point of evaporation in the leaf, there's a tiny meniscus that's forming. You guys remember what a meniscus is? Meniscus? Yeah. Meniscus. That little thing there. That meniscus, where evaporation is occurring, is creating water tension, right? That water tension here is actually being translated throughout the entire plant. It occurs here at the leaf, but remember that this is connected to the xylem here on, in the plant. And with hundreds, thousands of leaves, with thousands, millions of stomata throughout the plant, that's a massive surface area. And that huge surface area across the plant, pulling on that water, creating that tension, is literally pulling the water up out of the ground, down the water's potential gradient. Okay? So negative pressure gradient maintained by the xylem is pulling the water up. So here's what's happening. We've got more water in the ground than in the air. Water naturally wants to evaporate in that direction. Water enters the roots first because there's more solute in the roots than there is in the water. Water will naturally move down the water potential gradient because there's more solute there. Moves into the root. Then from the root, it goes into the xylem, okay? And then up here, at the end of the xylem where the stomata are, evaporation is occurring, and that evaporation is pulling those molecules out of the stomata, and it's pulling on the water in the xylem at the same time, okay? And it's that combined force of all that evaporation out of that stomata that's yanking that water up out of the ground. That's why it's called the cohesion tension theory. It's the cohesion of the water molecules sticking together, because water is a very cohesive molecule, all those hydrogen bonds, and the tension created by the evaporation from the stomata is pulling up out of the ground. Questions on that? Yeah.
So through the stomata is where the evaporation occurs?
Yep.
And then, so would the meniscus be convex to the leaf coming out of it?
No, it's inside.
It'd be coming in.
Right.
Okay.
It's, yeah, kind of.
Because I think it would be, Why? I mean, my brain would be coming out, like, cause then that would cause negative pressure if it was coming out. If it was coming in, would that be positive pressure?
I think it doesn't. I think,
Something happens.
I think what's important is the evaporation. I think what's most important is the evaporation, the actual shape of the meniscus. Because the problem is, it can't, so if this is the stomata, the problem that plants run into is it can't break this chain, right? If there's a break in that chain, so for example, if the meniscus went this way, I think it would break that chain. So I think the meniscus has to be like this. I think it does. Because you can't, because if you break the chain, it all stops. And that's what happens to plants when they freeze. Because if a certain part of the plant, tissue freezes, that water can no longer move. And then the water around it will keep moving, and you get these air pockets in there, and then the whole process stops.
What do you mean by chains?
It's one continuous flow of water from the root all the way through the plant out the end. It's those water molecules have to continue to be hydrogen bonded to each other. If you break that chain of hydrogen bonds, the whole process stops. Yeah.
And then the stomata is like a membrane,
The stomata is an opening to the outside.
an actual opening.
Yep. Yep, so that's where the evaporation's occurring, the water's leaving the leaf. Yeah. And the physical limit for this process is right at about 300 feet, about 100 meters. After that length, right, the literal weight of those water molecules will start to break those hydrogen bonds. So giant redwood trees are at about the physical limit of this process. They've grown as tall as physically possible. Yeah.
So if the stomata are closed,
Yep.
then there's no movement of water.
Right. Yep. If the stomata are closed, the water doesn't move. It just stays there. Yeah.
If the chain is broken, is it possible to remake that chain?
What's that?
If the chain is broken, the chain of like the water molecule.
Yep.
Is it possible to remake that chain?
Yep.
How is that?
Through, because there's tissue, because there's still water in the plant, and the plant's able to move that water around. That water will naturally diffuse on its own around. And there's this, let's see if I got it here. We're running out of time, but, oh yeah, here it is. So there's this thing called root pressure. And what happens is, so the solutes, so if you close the stomata, right, that is not allowing any evaporation to occur, but you still have the solute pressure between the root and the groundwater. And so the roots are still getting pumped full of water, and that water needs someplace to go. So what'll happen sometimes, this'll happen, you probably haven't noticed it. When plants first open their stomata in the mornings, all this water comes rushing out because the roots are just full of water. They've sucked up all this water. They open the stomata and all this water comes out of them. You may have seen it before and thought it was just dew. It may have just been dew. But that's what's happening here. So when there's enough water in the plant, in the roots, it'll push that out. Just push it, it'll fill in those gaps. Yeah. Okay. But the risk of breaking that chain is why the xylem in plants is very narrow, right? Because it's narrow, it allows those water molecules to stay close together and to maintain those hydrogen bonds. And remember, in dry climates like the Pacific Northwest, that's why we have trees like the conifers that only use the tracheids, which are small, more narrow than the vessel members. Vessel members are bigger. They're found in trees like the oaks and the maples, which you get on the East Coast, which is much wetter. So in a dry climate like the Pacific Northwest coast, you just get these tracheates, which are narrower because they don't want to break that chain. Okay. Question, Courtney? No? Any other questions? No? All right, I'll go back and cover this on Thursday. Where are we meeting for lab?
Put it on the-